And hello, everyone, for a short time of live Q&A and discussion. And thank you for being with us. I'm David Wold from TCAN with Bhavin Shah from Paramit. And you may notice, I'm pleased to introduce Noah Ditto from Carterra, Technical Product Manager, sitting in, pinch hitting this morning for Daniel, who could not be with us. And now I'd like to invite you to submit questions for any of us into the chat on your screen. We've already received a few questions to get us started. So while you're thinking and typing and sending those in, I will moderate or ask away. These questions so far are to you, Noah. So yes. What is the first one here I've got? What is the advantage to performing epitope characterization by SPR instead of plate-based ELISA assays?
Yeah, good question. I think really when Carterra's LSA technology came onto the market, it really transformed how epitope characterization worked in antibody discovery. Prior to that, ELISA was the workhorse based on its sheer throughput, which is needed in early discovery. But it always came with a limitation of poor resolution data, missing weak binders, sometimes challenges too with how you're labeling or otherwise attaching the antigen to the well plates in the assays. So HT-SPR, which is kind of the core technology that Carterra offers, allows you to do a lot more, get real-time binding profiles, get kinetic inferences from those interactions. And when you think about how ELISAs work, they're somewhat inefficient for this process where each well in a plate has to be compared with all the other wells. So for a panel of 96 candidates, that ends up being 96 plates. So it's a huge amount of plates to set up and manage and screen against versus on the LSA, we have a plate of antibodies that we put on the surface, a plate of antibodies that we inject, and a tube of antigen. None of them are labeled, so it's a very simple assay to run. And again, much as we've seen in the market, really has changed the adoption and approach to epitope characterization.
Thanks, Noel. Another question. Can I run large kinetic experiments, hundreds of clones, using supernatant antibodies against multiple targets? And if so, how much supernatant is needed?
Yes, the short answer to that is yes. Crude sources are a really common strategy for a lot of our customers. And that is because just there's so much upfront effort to invest into small-scale purification. When you go through that process, oftentimes early in discovery, there's just lots of candidates that fall out for various reasons. Bindings don't recapitulate. So really, everybody wants to screen early as they can directly in supernates and avoid any downstream additional processes that just incur more cost and resource overall. So on the LSA, it's quite a simple process. We capture out of crude material on the surface, and then we flow antigen. So in effect, the actual assay itself is in a purified state. To your second point on the quantity, that would be probably in the order of about 20 microliters or so. It's somewhat dependent on the expression levels, but as long as you have a reasonably good expressing system, you can assume that about 20 microliters is needed at most in order to dilute into the final assay concentration.
Thank you. Another question. How do you know how many epitope communities are appropriate for meaningful clustering?
Yeah, so there's a few rules that they're fairly straightforward to follow. I think sometimes when people start to do epitope characterization, particularly at the scale that we offer on our technology, there's a lot of data. So it can at times feel overwhelming. But if you keep a few basic rules in mind as you're analyzing the data, it's fairly straightforward. One basic rule is if antibodies block each other, it's most likely that they're epitopically related and therefore should maintain some level of clustering. And kind of following on to that, if you have antibodies that are competitive amongst each other, across different groups, that suggests that those groups probably have some shared relationships. They could be overlapping epitopes, for example, or otherwise somewhat structurally related in terms of where they're engaging. Those two kind of rules are fairly good to keep in mind, because really when we're doing competitive epitope binning, the blocking relationships are what drive our knowledge about how antibodies are related. If we fail to see blocking, we really can't say structurally how epitopically similar they are. But But the blocking relationships really do need to be kept in mind. If you see blocking between two antibodies, by and large, that's a really good indicator that you can cluster them with a reasonable level of confidence.
Hey, great. Got another question here, actually just popped in. Is it also possible or available in your system to screen using 1536 well plates?
Right now, at this time, we don't offer that capacity. Those are a little above the limit we have. So we do out of 384 well plates is the top capacity plate that we offer. But that does allow you to do to have up to 1,152 samples, as we have 384 well plate bays in the instrument. So you can approach that 1536 capacity in a single experiment by use of multiple 384 well plates on the system.
Good. And last one, at least so far, unless any new ones arrive, my lab doesn't have the capabilities to purify all our antibody candidates. Can I still get kinetics and epitope binning information on the LSA using crude samples? And how much, if so, how much supernatant would be required?
Yeah, definitely similar to what we the question asked before. So the both kinetic and epitope characterization start with the antibody on the surface. So we have a capture surface. It could be protein AG. It could be an anti-FC type molecule. We capture those antibodies out of the crude material, and then we have several standard off-the-shelf workflows where the customer goes through and can run kinetic analysis and/or epitope characterization analysis on that surface. And those are very popular. I think we initially launched our technology and didn't have maybe all those workflows in place, but in recent years have added them in, and they are quite popular, as I mentioned, for the same reasons that everybody wants to avoid an additional purification step if they can. So the LSA allows you to do that, going right into the analysis of your crude samples instead of having to source some purification process.
Awesome. Well, thanks for that answer and for all of these answers. That is the end and all of the questions that we have received, and also the last, pretty much out of time as well. So it's kind of perfect the way that's gone. Thank you one more time. Thank you, Noah and Bhavin, for appearing and being here today, and in the video with Daniel, and all of you for participating and connecting on the LinkedIn Live event today. All the discussion and questions were interesting as well. Please continue to send questions using the chat function on your screen. Any questions that we still or may yet receive after the event will receive offline replies. And lastly, if you found this interesting, please share it with your network, with your colleagues. And we hope to see you again soon at another TCAN LinkedIn Live event. Good day.
Bye-bye.
Bye.